Hello everyone, my name is Tanchanok Pangwatanasilikun from Mahidol University, Thailand. Today I'm a representative to present about Minerva mission, a CubeSat for demonstrating DNA damage mitigation against space radiation in silicons by using genetic modification. Space human exploration is a really intriguing idea in the space community. Mars missions are expected to be a big step towards deep space exploration in the future. However, there are many obstacles that make deep space exploration difficult, and one of them is space ionizing radiation. Space ionizing radiation causes dangerous health risks as it induces DNA lesions, double-strand breaks, and decreases the repairability of DNA. This leads to chronic health disease like cancer and apoptosis, which is the death of the cell. Now, what's our objective and how does it tie in with what I just mentioned? Our mission is to mitigate DNA damage against deep space radiation exposure by using genetic modification as an alternate solution. During our research, we found that the damage suppressor protein or DSUP found in target can prevent DNA damage from radiation and can tolerate up to 4,000 grays worth of radiation. When humans can only withstand 1 gray, we synthesize the DSUP coding gene imitated from the gene in the target and insert it into our model organism, the animal known as coelacanths. Unique thing about coelacanths is that they are able to survive in hibernation or without food for 4 months. This makes them suitable to be used in our mission as our payload will not need to contain a significant amount of supplies in order to keep the coelacanths alive. In addition, coelacanths have up to 83% of human homologous genes, suitable for representing human biological response from space radiation after genetic modification. What this means is that our CubeSat will be inhabited by an animal, something that is done incredibly rarely, making us thus much more different and unique from other CubeSats. Not only the subcoding genes are inserted into coelacanths, but green fluorescent proteins or GFP are also inserted along with the DSUB gene. The GFP serve a purpose of indicating whether the DSUB genes express their traits or not. During the mission, we will have 3 sets of coelacanths. 2 sets are in our payload and another group is set on the Earth as a reference. Transgenic coelacanths and space-controlled coelacanths are cultured in microfluidic shapes in the payload and will be sent out to the near rectilinear halo orbit to observe the effects from deep space radiation, which is the main factor of our experiment that we want to fulfill. Other than the previously mentioned about use of GFP, it can also be used to indicate the remaining DNA in coelacanths. DNA damage in neurons of coelacanths after absorbing space radiation will be observed by quantifying and comparing the appearance of co-expression GFP between wild-type coelacanths and coelacanths with DSUB. The left-hand side group is coelacanths before absorbing radiation, and the right-hand side is after absorbing radiation. If a large amount of GFP decrease after absorbing radiation, it means lots of DNA in coelacanths are damaged. To determine the enhancement of radioresistance in transgenic groups, we compare the remainder of GFP. If the remaining of GFP in the transgenic group is more than the space control group, that means the transferring can enhance radioresistance in coelacanths or the DSUB is work. Apart from the specifications of the Minerva mission I have mentioned, this secure CubeSat inhabited by an animal must have a lifetime of more than 4 months to obtain the results from the experiment. Total mass is less than 12 kilograms and the cost of this mission is about 1.2 million US dollars. This here is the system diagram of our satellite, and it contains all subsystems integrated into the satellite. However, I will only be going into detail on the payload aspect of the system, as that is our most integral part and is what makes us different from other competition. Our payload consists of an autonomous intelligent biological operating system which cultures silicons and observes the effects. Then the effects are analyzed with received radiation observed by the radiation dosimeter. Eyeball consists of 8 microfluidic chips with optical sensors, food pumps, and thermal control system. Optical sensors utilize multicolor imaging techniques to detect fluorescence. Optical sensors compose of near-infrared LED, which provide high resolution and high speed of silicon structure imaging, and blue LED, which detect the green fluorescent protein in silicons. And the last one is food pump, nutrients for a living. Because we want to observe biological effects from radiation in deep space, a radiation dosimeter is needed. We use a time-space linear energy transfer radiation spectrometer or LETS to detect the dose of galactic cosmic ray and solar particle events that silicons receive in near rectilinear halo orbit. Then, the received total ionizing will be calculated, collected into onboard storage, and sent back to the Earth. In order to let the CubeSat receive deep space radiation, the near rectilinear halo orbit is chosen as our destination. This orbit is a moon-centric orbit which expected for future cislunar space exploration since it was discovered in recent years. And to make sure that our CubeSat can rotate in this orbit, we simulate the trajectory of the CubeSat. The left picture shows the trajectory of the CubeSat by using the Moon as a center, and the right one uses Earth as a center. Then we calculate what trajectory path is most appropriate for the CubeSat. Apart from the trajectory simulation, the success of the mission is guaranteed by power budget, link budget, and volume budget calculations. The average and peak power consumption is not more than the maximum generated power. That means the maximum power generated can cover both average and peak consumption. In addition, we got 70% link margin for downlink and up to 87.5% link margin for uplink. The major volume of our CubeSat is the payload, and we also have margin for 19%. We plan to finish our preliminary research this year and do biological experiments next year. After that, we plan to develop a flight model from 2023 to 2024 and launch our CubeSat in 2025. If we can launch this project, it will provide many benefits in the field of space biology. IBO provides a platform for culturing animals in space and detect biological response from the CubeSat. If our method works, we can use this technique as a basis to develop future medicine to prevent long-term chronic health disease for astronauts. And if we can prevent the effects from space radiation, we can increase the feasibility for future space exploration. Apart from the benefits I have mentioned, Minerva is also in accordance with UN Sustainable Development Goals. The goal of UN SDG is about good healthcare and well-being, and the main objective of this project is to develop space medicine to prevent the effects from space radiation. Before we talk about another goal of UN SDG, Minerva project is launched by our space community called the International Society for Space Engineering in Life Science and Medicine. The society aims to lead young generation people into the field of space, provide space engineering education, and also space activities by collaboration with other societies from other different countries. Hence, the community activities follow the fourth goal of UN SDG, which mentions about quality of education. And here are our members who came up with this idea and tried to research and design the Minerva mission. Finally, we believe that our mission can make powerful impacts on deep space exploration, and we don't want to let our idea just be an idea. We will keep studying and make our passion drive the world. And that is the end of our presentation. If you have any question, don't hesitate to contact us. Thank you.